Hi everybody, Professor Gustin here, and this week we are going to be talking about biology of infectious disease. And I kind of like this lecture because it builds on some information that we've been learning this semester. We're going to talk about cells again and remind you a little bit about what we talked about cells earlier in the semester. And we're going to build on some of the information from the last chapter on DNA biology and mutations. And so, and natural selection and evolution, that comes in here too. So here we go. First one is right here. Remind yourself of the difference between eukaryotic and prokaryotic cells, or you might hear them called eukaryotes and prokaryotes. And this might sound familiar because we did this right at the beginning of the semester. We've talked a lot about cells. And the cells we've been talking about are eukaryotic cells, eukaryote meaning true nucleus. And remember that eukaryotic cells, humans are made of eukaryotic cells. They have a nucleus. Inside the nucleus, there's chromosomes. We talked a little bit about mitochondria, where the ATP is made. We talked about ribosomes, all these little dots where proteins are made, the cytoplasm, the boundary of the outside of the cell, the plasma membrane, and there were a lot of parts inside that we didn't talk about. But that's a eukaryotic cell. And at the time we talked about those, I also mentioned prokaryotic cells. Prokaryote means pre-nucleus. So these cells were actually around on the planet for more than 1 billion years before eukaryotic cells evolved. So these were around before a nucleus was a thing in a cell. For our purposes in this class, anytime I say prokaryote or prokaryotic cell, I'm talking about bacteria. Now there's a weird group of organisms called archaea that are very bacteria-like, but they are considered separate from bacteria that are also called prokaryotes. We will not be talking about those. So for our purposes, whenever I say prokaryote, think bacteria. And we all have some sense of what bacteria are. And we've also all heard of viruses. So we're going to talk in this lecture series, we're going to talk a little bit more about prokaryotic cells, the bacteria, and then we're going to have a real short discussion about viruses. But notice right here already, we notice the eukaryotic cells are quite large compared to the bacterial cells, which are still quite large compared to viruses. The next learning outcome says distinguish between pathogenic and non-pathogenic microorganisms. So I thought first maybe I better give you the definition of microorganism. And a microorganism is, micro means small, so it is a word that describes living organisms that are too small to be seen without a microscope. Microorganisms are living organisms that are too small to be seen without a microscope. But that definition also includes viruses. And I'm going to tell you right now that viruses are not living organisms. So the definition of microorganisms is living organisms that are too small to be seen without a microscope and lifelike entities. And other lifelike entities. That's kind of what you say. Viruses are kind of lifelike. So other lifelike entities. Okay. Microorganisms, living organisms that are too small to be seen without a microscope, and other microscopic lifelike entities like viruses. So we can say these microorganisms are pathogenic or non-pathogenic. And when we say they're pathogenic, we mean they cause disease. A pathogenic organism causes disease. A lot of bacteria cause disease, but actually most of them don't. And a lot of viruses cause disease, and some of them don't. So we have lots of pathogenic microorganisms, but we also have plenty of them, non-pathogenic, that do not cause diseases. So pathogenic means a disease-causing microorganism, and non-pathogenic is a microorganism that's unable to cause disease. And I just put this picture in here because you saw this earlier in the semester. Remember, here's some eukaryotic cells. These were cheek cells. And there's the nucleus where the DNA is. So I have 2 cheek cells here. And then all these little dark blue dots, they're bacteria, they're prokaryotes. And that's just to remind you that, oh yeah, bacterial cells are a lot smaller than eukaryotic cells. I want to just give you a general description of bacterial cell structure. So here's our eukaryotic cell. It's got lots of organelles. It's got a nucleus. Now we're enlarging this prokaryotic cell or this bacterial cell on the next slide. And what you can see in here is if we look deep inside, like I said, there's no nucleus, but it does have a chromosome. Remember, the human cell has 46 chromosomes, 23 pairs of chromosomes in the nucleus. A bacterial cell just has 1 chromosome, and that chromosome's in a circle. Remember, the chromosomes are made of DNA. DNA is the molecule that carries all the instructions on how to make the cell. So bacteria are a lot simpler than eukaryotic cells, so they don't need as many instructions. So they just have 1 chromosome that has all the genetic information on it. You see some other structures here related to prokaryotic cells. You see ribosomes. Again, remember, ribosomes are where the proteins are made. But there's no mitochondria. There's none of these other, when we look at the eukaryotic cells, we see a lot of other organelles that we didn't talk about. Prokaryotic cells don't have those. They're real simple. They just have the chromosome and the ribosomes. They also have a plasma membrane. Remember, the plasma membrane was the phospholipid bilayer that controlled what went in and out of the cell. And so bacterial cells have a plasma membrane. All cells do. And bacterial cells also have a cell wall on the outside. Now, you might have remembered we talked a little bit about this. We said plants have a cell wall. In fact, that's the fiber that we eat. When you eat plants and that crunch you get, some of that, well, it's actually mostly the water that's making it crunchy. But plants do have a rigid cell wall around each cell. Animal cells don't have that. You can see back in our eukaryotic cell here, this is an animal cell. It's kind of soft. We got the plasma membrane on the outside, but no cell wall. But bacterial cells have a cell wall. There you go. And it's got some other parts too. But for our purposes, we're not going to talk about anything else. But I do want you to recognize that there's, describe and recognize the 3 common shapes of bacteria. So when you look under a microscope and you see bacteria, here I see lots of little circles and a chain there. Oh, there's some circles clustered together. There's actually 3 major shapes of bacteria. And they're called bacillus, coccus, and spirillum. A bacillus-shaped bacterium is kind of rod-shaped. So this looks like, this is a picture that was put together from some electron microscope pictures, highly magnified. And you can see here some prokaryotic cells that are bacillus-shaped. And even in our example cell that we've been looking at, that's a bacillus-shaped bacterium. Coccus-shaped bacteria are round. There's some coccus. And spirillum have some kind of curlicue or curvy. If we look back at our cheek cell, here's our cheek eukaryotic cells, and here's some bacteria on there. If you look really carefully, I don't know if you guys can see it, but I can see it pretty well, that there's 2 coccus, there's 2 round bacteria together. That's a lot of these look like, oh, there's 1 coccus. Some of these might be bacillus shaped. There's one that might be bacillus, or they might be 2 coccus together. I can't really tell. But so bacillus means a rod shaped or elongated. Coccus means round, and spirillum means curvy. And I just wanted to show you a couple words. You do not have to remember these, but notice that some bacteria have a little bit fancier names. And if you're called streptococcus, so coccus means round, strepto means chains. So this bacterium right here is streptococcus, chains of round bacteria. If you're called staphylococcus, it means you're clusters. See how they're grouped together? Coccus means round. There's a group of round bacteria. These are staphylococcus. Later in this lecture, we're going to talk about Staphylococcus infections. And so if you look at that bacteria under the microscope, they're going to be round and they're going to be in clusters like this. And so they got named after what they look like. Just like you've probably heard of strep throat. Strep throat is caused by round bacteria in chains. And if you looked at them under the microscope, they'd look a lot like this. So sometimes we name bacteria after what they look like under the microscope. Just a little bit on how bacterial cells divide. They divide by something called binary fission. So when we say bacteria are growing, or we say, oh, the bacterial, we've got an infection, the bacteria are growing. We don't mean one bacterial cell is getting bigger and bigger and bigger. We mean it's dividing. It means that one cell is dividing into two cells are dividing into 4, and so on and so on. So we call this cell division, we call it binary fission. So we start off with one bacterial cell, and then it copies its DNA. That's what this red part is. That's where the chromosome is. And in this picture, don't let the word nucleoid bother you. I told you that bacteria don't have a nucleus. Nucleoid just means the nuclear area where the chromosome is. But so you copy the DNA, and the cell starts pinching into 2 cells. And so one bacterial cell will divide into 2. 2 will divide into 4. 4 will divide into 8. So when our bacteria are growing, they're growing in numbers by binary fission. So if I have 1 million bacteria, it takes it just as long to form 2 million bacteria as it takes for one cell to divide into 2. Okay, if each cell is dividing at the same time, 1 million bacteria can make 2 million bacteria at the same speed that one cell can divide into 2. And there's a little video in your book that you might want to click on the link. It's in the reading part of the book in the chapter. You might want to click on it. Tux just mentions binary fission. I also wanted to just give you a little bit of information about viruses. And so let me go back to our picture at the very beginning. Remember we said we're going to talk about eukaryotic cells and prokaryotic cells and viruses, which are not cells. They're not living, but they're lifelike, and they can infect things, and we do consider them microorganisms. And we've all heard of viruses, but I think it's really easy to mix up bacteria and viruses. So it says, describe the general structure of a virus. So think about what you know about cells and compare that to a virus. Already we know that the virus is a lot smaller than the eukaryotic cell or the prokaryotic cell. So viruses are a lot smaller than cells. And here's a cartoon of a certain kind of virus. And that might even be an HIV virus. I'm not sure. Pretty sure it is. But here's this kind of weird looking thing. And inside, it has DNA or RNA. It still needs instructions on how to make a virus. It needs a nucleic acid. But we said all living cells have DNA, and they use the DNA to make RNA, and the RNA is used to make proteins. We talked about that last week. But viruses, they don't all have DNA. Some only have RNA. So viruses can have DNA or RNA, and it just depends on the virus. So I think that's what they're showing you here. There's some, in this particular one, they're showing, this is the RNA. They also have a capsid. And this picture right here, you can see this is a virus that has DNA. So there's some DNA, and all these little green dots on the outside make up what we call the capsid. The capsid is the protein coat. A capsid is a protein coat. So the minimum thing the virus can have is some nucleic acid, some DNA or RNA, and a protein coat called a capsid. And that's all some viruses have. They don't have ribosomes, they don't have a plasma membrane, they don't have mitochondria, they don't have any of that. They don't have a cytoplasm, they don't have a nucleus, nothing. They've just got these chemicals. Some viruses have a layer on the outside called an envelope. And so they're showing you that in this example, where the green here is the capsid. And then this lighter layer out here is the envelope. So some viruses do have another layer on the outside. That's why these two can look so much different. And a lot of viruses have these things on the outside called spikes. And the spikes can help them hook up to others, hook up to cells, and I'll show you that in a minute. Spikes can help them hook on to what we're going to call a host cell. So I showed you how a really simple example of how bacteria divide. They copy their DNA and they divide into two. But viruses divide in a different way. And it says, explain how viruses divide and what it means to be an intracellular parasite. So it turns out a virus must have a host cell in which to multiply, which means a virus, here we go, a virus needs a cell. A virus needs a eukaryotic cell or a prokaryotic cell, and it multiplies inside of that cell. Every kind of organism on this planet can be the host to a virus. Viruses infect every kind of cell we know. Bacteria can have viral infections. In fact, they're very common. So even bacterial cells can be the host to a virus. Because of this, we say viruses are obligate intracellular parasites, meaning obligate means it has to, intracellular, be inside of parasite living off of another organism. So viruses have to go inside of another organism in order to multiply, in order to make new viruses. A virus has to go inside of a living cell to make new viruses. That's really different than bacteria. We said the bacteria can just divide into two. You give it enough food, you give it moisture, and they can divide into two. A virus needs to find a cell, and it could be a bacterial cell, it could be a human cell. But the other thing to be aware of is they only multiply in specific types of host cells. One of the things we're going to talk about is we're going to talk a little bit about COVID and pandemics and the COVID virus. Well, we all know cold viruses infect the cells in your head. When you get a cold, it's in your head. It's in your nasal cavity. COVID infects the cells in your lungs. So viruses will only multiply in a specific type of host cell. And that host cell even it has to be a certain kind of organism, like a bacterial virus will not infect humans, or it can be even within an organism, a specific kind of cell. Cold viruses only affect the cells in the upper respiratory tract. COVID only infects cells of the lower respiratory tract. So if we just take a quick look at this virus, here's a virus, and here's some kind of, looks like a eukaryotic cell because it has a nucleus. Here's the virus using its spikes to find a molecule on the surface of the host cell. And it says, oh, I match. I want to infect that cell. So it says the virus attaches to the host cell. It goes inside. It's showing you it's going inside. And then what it does is now we have the viral DNA. In this case, it's viral RNA inside the host. And it uses that to tell the host cell to make viral parts. And so there's a lot more going on here, but here I'm just going to walk you through what I want you to know. The virus attaches to the host cell, it goes inside, it releases its DNA or its RNA. In this case, it's going to have some viral RNA. It's going to use that, it's going to use the ribosomes from the host, it's going to use the ATP from the host. All the building blocks are from the host cell. And it's going to make viral parts. And then so that's called biosynthesis. And then it's going to assemble them all together and make viruses. And the viruses pop out of the host cell. And that eventually will kill the host cell. Okay, so a virus is going to make new viral parts inside the host cell. I think about viruses like a kid living with their parents. A kid doesn't need its own house. They don't need their own money because they're living off the parents. They don't need their own furniture because their parents have furniture. So the kid is like the virus and the parents are like the host cell. The host cell gives the virus everything it needs to make new virus. Everything except for the DNA or the RNA. And I think there's a little video that you might want to check out in the ebook on how viruses divide. And I'm going to stop right there.